If you want lunch, we have to sit down, please, ladies and gentlemen. Yeah, I'll announce Susanna, yeah.
You can announce Susanna.
Oh, you? Oh, sorry, Jim, I didn't read the colour code. You do it. It's right, no, it's fine.
Could
sorry,
you please take your seats? Welcome back. The President and Professor Suzanne von Kammerer will now present the 2017 Fenner Medal.
Thank you, Jim. The 2017 Fenner Medal recognises outstanding contributions to science by the late Professor Frank Fenner, AC, CMG, MBE, MD, FAA, FRS. Its purpose is to recognise distinguished research in biology, excluding the biomedical sciences. The recipient of the 2017 Fenner Medal is Professor Simon Ho. I invite him to come up to the stage, and then Professor Susanna von Kammerer will read the citation.
Professor Ho has transformed the use of molecular clocks in biology, a way of estimating evolutionary rates and timescales from DNA sequences using statistical models. Professor Ho's most important and influential work has been on models of evolutionary rate variations through time. His research has critically changed the way in which biologists use molecular clocks, especially when studying the timescales of recent events in evolution and human prehistory. This has important impacts on a broad range of studies in conservation genetics, speciation, diversification, domestication of animals and plants, and the population dynamics of pathogens. Professor Ho, congratulations, and I invite you now to address the meeting on the evolutionary timescales of the tree of life.
Okay, thanks very much. I'd like to start by thanking the Academy for this award. It's a real honour, and it's a real pleasure to have this opportunity to talk about my research to such a distinguished audience. So my research over the past few years has really focused on estimating evolutionary rates and timescales, and we can do this using a statistical tool known as the molecular clock. And so much of our understanding of the evolution of life on Earth has been informed by the fossil record, but the fossil record is largely silent on the evolution of a number of groups. For example, microbes like viruses and bacteria have left scant traces in the fossil record, as well as larger soft-bodied organisms like flatworms, and even larger organisms like birds, which do have hard body parts. Some lineages of birds have a very incomplete record in the fossils. And as we look deeper back in time, the fossil record becomes increasingly incomplete, and this leads to substantial gaps in our understanding of the evolution of life on Earth. And one way to fill some of these gaps is to analyse the DNA from modern organisms and reconstruct the evolutionary timescale using molecular clocks. So the fundamental principle behind molecular clocks is fairly simple. I'll illustrate this with an example from 4 primates. So let's say we have DNA sequences from these 4 primates: human, chimp, gorilla, and macaque, and we have some estimate of the divergence time between humans and chimps based on the fossil record, 6.5 million years. This reference point allows us to infer the rate of evolution along the branches in the human and chimp group. And if we assume that the evolutionary rate has been relatively constant throughout the primate tree, then we can also infer the times of divergence of these other events in the tree between humans, chimps, and gorillas, and with macaque. And we can then rescale the tree so that it's proportional to a time scale measured in millions of years before present. So in this procedure of molecular dating, there are a number of very important sources of error. And the first of these is the uncertainty in the fossil that's used to calibrate the molecular clock, the uncertainty in the estimate of the evolutionary relationships among the species that we're studying, and also uncertainty in the model of rate variation across branches in the tree of life. and there's been significant progress in all of these areas over the past decade or two. And much of my research focuses on the methodological development in all three aspects of the molecular clock, but I won't go into the technical details today, but instead I'll focus on three case studies from my recent research using molecular clocks. And so these will involve the evolutionary history of flowering plants, of modern birds, and of Australian marsupials. So when we look at the evolution of plants on Earth, first few 100 million years, plant diversity was dominated by these two groups, pteridophytes, or ferns and their relatives, and by gymnosperms, which are conifers and their relatives. And this plot shows the number of species through time inferred from the fossil record. In the late Mesozoic, we see the origin and diversification of angiosperms, or flowering plants, and flowering plants now dominate the species diversity of the modern flora. So my colleagues and I sequenced chloroplast genomes from a number of flowering plants and assembled a dataset that represented many orders of living flowering plants. And when we analysed these data, we inferred the evolutionary relationships and the evolutionary timescale, and we found a much more protracted evolutionary history than suggested by the fossil record, with an origin of flowering plants in excess of 200 million years ago. And this converges with recent molecular estimates based on smaller data sets. And the implications of this are important for how we interpret the evolution of flowering plants alongside other groups such as the insect pollinators. And moving on to birds. Birds are a group that have a very long evolutionary history. Many of you will recognise the fossil on the left there, which is Archaeopteryx, generally recognised as the first true bird. And there are also more recent bird fossils, such as Confucius ornis, shown on the right there. That's one of the most common fossils in the record. Several hundred specimens are available. But it's not until much later that we actually see the first evidence of modern groups of birds. And these blue bars show the known fossil records of a range of modern bird orders. And a lot of these fossil records commence within the last 60 million years. And this led to the idea that the extinction of the dinosaurs, which occurred about 66 million years ago, opened up a range of niche space, allowing modern groups of birds to diversify. And so I've had the privilege of being part of the Bird Genomics Consortium, which is an international team of researchers. A few years ago, we sequenced the genomes of 48 different birds, representing all of the modern orders of birds. And we inferred this evolutionary tree, or phylogeny, using over 8,000 genes. And we inferred the timescale of evolution, and what we found was that many of the divergences among orders of birds occurred in a very short space of time, around the time of the extinction of the dinosaurs. And that lends support to the idea that the bird diversification was tied to the extinction of the dinosaurs. And the eventual goal of this consortium is to sequence the genomes of all 10,000 living bird species, and that will bring some very significant computational challenges. And my final example brings us back home. I'm part of the Mammal Genomics Initiative, and the goal of this team, which includes representatives from universities and museums throughout Australia, is to generate large amounts of genetic resources for all of the terrestrial mammal species on our continent. And this fauna is dominated by bats, by rodents, and marsupials. And as the first stage of this project, we analysed DNA from marsupials, representing all of the modern families of marsupials. And based on an analysis of 1,500 genes, and we developed a range of new methods to handle the heterogeneity in the signal across these genes, we inferred the evolutionary relationships and timescale of these marsupials. And we found some surprising results, one of which was the close relationship between the enigmatic marsupial mole and bandicoots in the bilby. And we found that some of the key divergences among marsupial orders occurred during a time of major transition in the Australian environment and climate, when the continent was becoming increasingly covered by rainforest. So these particular examples relate to some very well-studied and well-understood groups of organisms, but we can also apply molecular clocks to other branches of the tree of life, including recently evolved pathogens, viruses, bacteria, on very short timescales, Molecular clocks are constantly being refined. Some of my active areas of research include developing methods for handling genome-scale data, for improving the reliability of methods for estimating evolutionary rates, and characterizing rate variation across the tree of life. And so this work was done in collaboration with a very large number of researchers, including those involved in the B10K Bird Genomics Group and the Oz Mammal Genomics Initiative. And a lot of the hard work was done by members of my research group, shown in annual snapshots here over the past 6 years. And our research has received funding over the past decade from the Australian Research Council. Thanks for your attention.
Time for some questions? Yeah. There, then there, then there.
Roger Short. Could you tell us whether the asteroid impact in the Gulf of Mexico at Chicxulub, did that create any extinctions other than the dinosaurs?
Yeah, quite a wide range of extinctions in various groups, dinosaurs being the most well-known, but certainly a range of other reptiles and a lot of invertebrates as well. Yes.
I may not have quite understood what you did, but it sounds as if you focused on genes rather than intergenic space. And do you think that could have introduced any biases into your trees?
It is possible because genes are under different forms of selection, selective pressure, selective constraints in different organisms. But when we're looking at these sort of timescales, we need to look at coding regions because we can be more confident in the homology across organisms. So in terms of aligning the sequences, using the exons or the genes is probably the best source of information.
Sam Berkovic, University of Melbourne. Thanks for a lovely talk. How comfortable is the assumption that the rate of mutation has stayed more or less the same through geological time, given changes in temperature, ionising radiation, etc.?
There are a lot of factors that are known to affect rates of evolution, including mutation rates and longer-term evolutionary rates. Actually, the rates of spontaneous mutation are poorly understood. There are only a few estimates from vertebrates, and that's something that we're trying to change. I'm working with collaborators in Japan to sequence genomes from family trees of different organisms. But we know that differences in generation length, in longevity, in exposure to mutagens will all affect these rates of evolution. So at the moment, we don't really have any reliable mechanistic models of rate variation. They're typically statistical models that just capture the variation in rates.
One last question.
So there's a tremendous field that you work in, Joss Hawthorne, University of Sydney. Does the method that you use rely on slow progressions that you can identify most easily, or do you ever see evidence of a dramatic mutation across many branches?
We typically select data that are appropriate for the timescale of that we're studying. So if we're studying recent events, we look at rapidly evolving sections of the genome because when too many mutations occur in a particular section of the genome, then the signal will be saturated and there'll be too much noise relative to the genuine evolutionary signal. So it's largely a matter of data selection, but we also have models that account for that hidden mutation.
Thank you very much, Professor Hope.
The 2017 Gottschalk Medal. The Gottschalk Medal recognises outstanding research in medical science and honours the contributions to science of the late Dr. Alfred Gottschalk, a Fellow of the Academy. Its purpose is to recognise outstanding research in the medical sciences by researchers up to 10 years post-PhD. The recipient of the 2017 Gottschalk Medal is Associate Professor Catherine Elizabeth Holt, and I invite her to come to this.
Associate Professor Holt's current research tracks the evolution and spread of deadly infectious diseases and the development of antibiotic resistance in Australia and the developing countries. Her in-depth studies on the evolution of specific pathogen populations use the most advanced DNA sequencing technologies that allow detailed comparisons of the genomes of hundreds of closely related isolates of some of the pathogens. These have revealed how pathogens are evolving in response to exposure to antibiotics, vaccine-induced immunity, and natural host immunity. Her work has provided important advances in understanding disease transmission, control of infection, and informs public health policy in practice. Associate Professor Holt, congratulations, and I invite you to present to us regarding microbial genomics and antimicrobial resistance.
Thank you. It's a great pleasure and honour to be here today to receive this award. So I work on bacterial pathogens that cause infections in humans. And this is obviously important. We've heard a bit about this already today. But over the last 75 years since we've had antibiotics to treat infections, the rate of deaths from infections has decreased dramatically. However, the bacteria are fighting back and evolving drug resistance, and we have seen pathogens with resistance to all classes of antibiotics that are currently available for treating infections in humans. Because of this, infectious disease is still a major problem affecting global health. It's the #1 killer of children globally, and closer to home, 1 in 10 hospital admissions result in infection. So I use genomic approaches to study this problem. This generally involves working with clinicians and public health labs who isolate bacteria from patient specimens. We then take those pure cultured bacterial isolates, extract DNA and sequence their genomes. And then where me and my lab spend most of our time is on comparative genomic analysis of the resulting DNA sequences to investigate questions relating to transmission and evolution of the bacteria and their antibiotic resistance. So I'm just going to show you one example of this today, which is my favourite bacteria, Salmonella typhi, which causes typhoid fever. So together with colleagues in the International Typhoid Consortium, we sequenced 2,000 Salmonella typhi isolates from 63 countries across the world. And the phylogenetic tree on the left shows you the genetic relationships between these strains, which reveal the population structure of the pathogen globally. The map on the right shows you the distribution of these lineages, which are each indicated in a different colour, across the world. And in each region where typhoid cases occur, multiple different lineages circulate. Now, I'll draw your attention in particular to the red clone here, which we call the H58 clone, which accounts for about half of all of the isolates in this tree. And this clone is associated with multi-drug resistance. So this just shows you the distribution of this particular antibiotic strain of typhoid across the world, and it is now common in all regions where typhoid is endemic. So we have used this genome data to try and understand how and where this clone has emerged and how it has evolved drug resistance. So using the molecular clock techniques that Simon Ho has just so beautifully introduced, and also the geographical information, we have determined that this particular clone originated in India around 25 years ago in the early 1990s and has since spread across Asia and Africa with multiple separate introductions into Africa where it is now continuing to spread. And one of the reviewers of this paper noted and wrote a commentary article on the fact that this map of the spread of this antibiotic-resistant clone mirrors very closely the patterns of human migration out of India. And typhoid is a human-restricted pathogen. Humans are the only known reservoir for this bacteria, and it's certainly true that human movement is the major driver of spatial dissemination of this pathogen. So we next wanted to understand how typhi actually evolved to be so drug resistant. And what I'm showing you here is just a genetic map of all of the resistance genes that we found in this antibiotic-resistant clone. All the arrows are different genes. The yellow arrows are antibiotic resistance genes, and they together encode resistance to these drugs that are labelled in red, which include all of the first-line drugs used to treat typhoid. around the world. Now, these cluster together in a single gene cluster, which is a mobile piece of DNA. And what has happened in the case of typhi is that this group of resistance genes has transposed into a plasmid, another form of mobile DNA, which has then, at some point in recent history, around about the early 90s, met up with a typhi cell. The plasmid encodes all of the proteins that are required to build a bridge into neighbouring bacterial cells and to pass the plasmid and its drug resistance cargo into the typhi cell. So this plasmid, with its drug resistance genes and the typhi chromosome of the particular haplotype H58, have been circling, spreading across the world together for the last 25 years or so. Now, more recently, we have seen that this cluster of resistance genes has transposed into the chromosome, allowing the cell to drop the plasmid and its associated fitness costs and any potential risk of actually losing these resistance genes. So we are now believe that these resistance genes are going to be a permanent fixture of this particular clone, which is severely limiting treatment options. Now, I just wanted to pause for a second to say that working out these these complex resistance elements is difficult to do. Ruth Hall, who is here and a member of the Academy, has spent much of her career working out the fine detail of these elements. And in my group, we've spent a lot of our time developing new bioinformatics software tools to try and do this in a high-throughput manner using high-throughput genome data, where we've got thousands of genomes to sort through in one go. And these are just some examples of tools that we've had to develop in order to do this work in collaboration with people like Ruth and Mike Inouye. Just switching back to typhoid, what I've been telling you about is how we've been using genomics to reconstruct the past and understand how resistance has evolved to get us to where we are today. This same genome data now provides us with a genomic analysis framework to support ongoing global genomic surveillance of typhoid fever around the world. We're working with collaborators at the Sanger Institute to make this available in an online platform that's easy for other researchers and public health labs to use to try and track and understand ongoing and emerging problems in their local endemic areas. I also just wanted to mention that I'm working with multidisciplinary teams using genomics to investigate typhoid infection and transmission in various endemic regions around the world, hoping to inform the use of of vaccine strategies against typhoid in endemic areas where they're most needed. And lastly, just to note that while I've had to pick my favourite organism to tell you about today, my group and my collaborators spend a lot of time working on other bacterial pathogens of global health importance, including diarrheal pathogens, tuberculosis, which we've heard a bit about today, and hospital-associated infections where the drug resistance burden is particularly high. So with that, I'd just like to acknowledge my key collaborators on the typhoid work, Stephen Baker at the Oxford Unit in Vietnam, and Gordon Dugan, Vanessa Wong, and my other colleagues in the International Typhoid Consortium, and my research group at the University of Melbourne. So the science that I've presented today is very much team work, and I have the pleasure of working with a fantastic internationally dispersed team that make this research as fun as it is fascinating. Thank you.
We have time for questions. Are there any? Over there, Jenny.
Thank you. Jenny Graves, La Trobe University, Melbourne. You mentioned fitness costs, and that's something I've always been interested in. I believe that if you, I've been told that if you grow these multi-drug resistance strains in the absence of drugs, that they'll eventually lose the resistance. Is that just because they're on a plasmid, and once they're incorporated into the chromosome, are they quite stable, or are they also lost in the absence of continued selection?
So, like most of biology, this is very messy and there's no one rule. In areas where the plasmid, in Southeast Asia, when this plasmid was first noted in the late 90s as causing a severe problem. The WHO changed their recommendation to no longer use the first-line drugs whose resistance was encoded on the plasmid and to use fluoroquinolones instead. That resulted in the loss of that plasmid from that area. But wherever those drugs are still used, the plasmid remains, and as soon as you reintroduce using those drugs, the few strains that were still hanging around with the plasmid come back to the fore and take over. So there is certainly a fitness cost associated with this particular plasmid. Other drug resistance mutations, even just looking at typhoid, other drug resistance mutations, including those for fluoroquinolones, seem to carry no fitness cost at all. In fact, in the lab, they actually make the strains more stable at replicating and can actually outcompete the wild types. So we don't see reversion of those mutations at all. And at the moment, the most concerning thing about this recognition that the resistance genes have moved into the chromosome is that we don't think that there's any fitness cost associated with maintaining those genes at all. So we now have strains that have all of those resistance genes for first-line drugs, plus mutations conferring resistance to fluoroquinolones, and all we have left are third-generation cephalosporins, and there are now outbreaks of cephalosporin-resistant typhoid happening.
Last talk, Catherine. Have you considered incorporate antibiotic usage into your bioinformatic analysis? Do you think that will affect the spread of certain clones?
Yes, so as I've just been mentioning, we can see the effect of different usage patterns on changes in the resistance elements, whether they're acquired genes or mutations. The problem is that a lot of people don't keep good records of what they're treating with, and a lot of the typhoid endemic areas that we're working in, particularly in Asia, in Vietnam and in Nepal, people just use leftover drugs. We've done a healthcare seeking behaviour survey and found that that's often the most common thing that you do when you think you've got a fever. So we don't actually have great drug usage data for most locations, but I think it's an incredibly important thing moving forward that antibiotic resistance surveillance includes both surveillance of drug usage and resistance in the pathogen populations.
Thank you very much, Professor Hopp.
The 2017 John Booker Medal in Engineering Science recognises outstanding research in engineering mechanics and related fields within engineering and applied mathematics disciplines. It honours the memory of Academy Fellow Professor John Robert Booker. The recipient of the 2017 John Booker Medal is Distinguished Professor Dae-Yong Jin. I invite Dae-Yong to come to the stage and for Vice President Professor Jim Williams then to read his citation.
Professor Jin is a leader in engineering time-resolved photonics devices and luminescent nanoprobes which can upconvert low-energy infrared photons into more useful visible light for high-contrast detection. While his research opens up many opportunities in biomedical devices, early diagnosis and light-triggered nanomedicine, his nanorods can also be made into an invisible ink to protect pharmaceuticals, medical courier supplies, passports, banknotes and more. Professor Jin, congratulations. I invite you to present your talk entitled Photon Upconversion Superdots, Lighting Biomedical Devices.
Thank you, Jim. Feel deeply honored to receive John Booker Medal for this year for engineering science. Before I start my scientific presentation, I would like to acknowledge the Australian Research Council for funding my research for the last 7 years, and also my industry partners who have been motivating our research so that we aligning our daily research activity towards solving some real-world problems. And of course, a fantastic team of my PhD students and postdoc at both the University of Technology, Sydney and Macquarie University, and all my collaborators at different countries, interdisciplinary from different interdiscipline to contribute to this work, and which is really teamwork that enable the research outcome I'm going to present to you today. So when I start my PhD at Macquarie University with Professor Jim Piper, I was challenged by this single problem in early diagnostics. We found, detecting and finding this needle in the haystack in biological sample holds the real promise to enable the very early stage diagnosis of cancer, detection of waterborne, foodborne pathogens, and detection toxins and contaminations. So as a physicist by training, I start to summarize this biological challenge into 4 parameters, which is sensitivity, speed, throughput, talking about how possible we can detect multiple analytes in a single test, and also resolution, including the spatial resolution and temporal resolution, so that as a tool developer, we can provide more detailed information to biologists to understand the life sciences and the cause of diseases. Immediately after my PhD, I realized this first technology to reduce all the autofluorescence background by engineering this very low cost but very high contrast fluorescent microscope. You can see this image showing here is actually a giardia sitting in a very big matrix of water dirt concentrate. So the technique itself can reduce all the autofluorescence so that we can directly see the single giardia cell without any background interference. So the mechanism is quite similar, actually is inspired by a scenario when we sit at home watching TV in the evening, and suddenly the house is powered off, and everything else will go to darkness, but only the TV screen will continue to glow for a little while. So in biology, we can use this long lifetime luminous probe to label this single cell, and then we can use a pulsed illumination and use the optical chopper to synchronize with the pulse. So only when the pulse finishes, the chopper blade move away, so allow you to have a detection. So in this case, you can detect a single cell at very sensitive level. After this work, I've been working with Olympus Australia for a number of years, and through this PhD project, Li Xin, who finished her PhD and translating this technology into a touch-on, plug-in and play module, so that we can provide much broader access for biologists to use this powerful technology for background-free sensing. So Li Xin finished her PhD 2 years ago and start the research engineer work at Olympus Australia. Now she's looking after all the high-end fluorescent microscope in this country. So the next breakthrough we did, again, is inspired by industry collaboration with BD Biosciences in San Diego many years ago. And that time, BD is the #1 provider for flow cytometries. And they challenged us by developing a technology to detecting a very low abundant biomarkers on the surface of cancer cell. In this case, is a leukemia cancer cell. And when the number of antigens are getting really, really small, and then we found, you need an amplification strategy. So we start to think about nanotechnology to amplify your signal level, because each single nanoparticle, you can encapsulate multiple emitters, so that you can use a single nanoparticle to recognize each single biomarker on the surface of the cell. So through this experience, we start to love nanotechnology, and we start to focus our own research to develop a super bright nanoparticles, which the goal is to increase the number of emitters per nanoparticle from tens of them to hundreds of them and to thousands of them. But the dream has been stopped by a limitation called concentration quenching in physics. So that means once you put too many emitters within one nanoparticle, the emitter will start to cancel each other, rather than to getting the nanoparticle getting brighter, but the nanoparticle getting dimmer. So what we found here, for the first time, we found concentration quenching actually is power dependent. So that means if you give a high concentration pumping energy, so you can build multiple emitters in single nanoparticle. So by this way, you can use a nanoparticle focusing on a microscope, so that you can develop a super bright nanocrystal for sensing. Again, through collaboration with Tanya Monroe at University of Adelaide, we challenged the detection limit in fiber sensing. So in this case, we can improve the sensitivity by 3 orders of magnitude until we can see single nanoparticle. Okay, in the same time, we try how to using this different lifetime decay rate and have different thin size of nanoparticle so that they have a different decay and take turn to shine. And we can use this decay rate for multiplexing applications. So in biology, you can see, sorry, this stopped. So in biology, you can use this different lifetime decay rate to simultaneously detecting multiple pathogen DNAs in a single test. So this is holds a good promise for detection of infection disease. Oops. And last month, we report this new result by finding a new physical parameter from superdots. So we can use a very low power super diode laser to replace the traditional big power laser in super resolution microscopy. So this will hold the real promise to translate the super resolution nanotechnology, nano microscopy technology for biological imaging applications. And then this is another exciting project with a Deming who's synthesized nanoparticle for about 600 times. And we know how to do the 3 controlled synthesis of single nanoparticle with different shape, different morphology, but have multiple functionality. So we can use this technology for simultaneously realize full modality imaging in biomedical engineering. So I would like to use this short perspective article I wrote for Nature Photonics last year to emphasize how important it is to stimulate the international, interdisciplinary, and industry collaborations to really push the translation from material to device integration. So finally, thank you for all.
Questions or comments? Over here, Bob.
Well, Bob Williamson, Melbourne. Very impressed at the extent to which you can isolate the very rare cells that occur in the circulation. The alternative approach, as you know, is there's a lot of DNA that is floating free in the circulation. Would you like to discuss the advantages and disadvantages of looking for these very rare cells as compared to going for the DNA that's there in plasma?
Okay, this is a very good question. I agree with you. There's multiple pathways for the biomarker detection. And, you know, I keep saying, you know, none of the single technology can solve the problem. The only solution will have different technology to work together and collaboratively to solve this problem. So we can, at this stage, we have 3 pathways to detecting biomarker. One is detecting directly label the cell and separate the cell, as you mentioned. Second is directly detecting the DNA analyte by the beads assay, by flow cytometry beads assay as well. Also, the third way we found recently very interesting to detecting exosome. So that's the tiny nanoparticle containing all the biomarkers in the circulation. So we are working very hard towards, you know, almost parallel and collaboratively using these 3 different strategies to get much low concentration detection.
Any further questions or comments? Well, let's thank Professor Jin again.
The 2017 Moran Medal recognizes the contribution to science of the late Patrick Alfred Pierce Moran, FAA. Its purpose is to recognize outstanding research by scientists up to 10 years post-PhD in the calendar year of nomination, except in the case of significant interruption to a research career, in one or more of the fields of applied probability, biometrics, mathematical genetics, psychometrics, and statistics. The recipient of the 2013 Moran Medal is Associate Professor Joshua Ross, and I'll invite him to come to the stage, and then Professor Sue O'Reilly will read the citation.
Associate Professor Ross has made important and influential contributions to applied probability and statistics and through application to conservation biology and public health. His research has focused predominantly on addressing problems arising in infectious diseases, epidemiology and conservative biology, though the methodological developments that he has provided to solve many of these problems are very widely applicable. These application topics are of great importance. His contributions to these fields are very significant. Associate Professor Ross, congratulations, and I invite you to present your talk entitled Characterising the Epidemiology of Pandemic Influenza.
Yeah, I'd like to start by thanking the Academy. It's obviously a tremendous honour to receive an early career medal, and I'm delighted to be here. Before I go into talking about one example of the research I do on characterising the epidemiology of pandemic influenza, I thought I'd start by trying to characterise my own research and the research of the group I'm in at the School of Mathematical Sciences at the University of Adelaide. I guess there's four kind of key characteristics that it has. One is that I think the problem needs to have practical importance. So during my PhD, I looked at questions in conservation biology. For a long time now and continuing now, I look at the spread of infectious diseases and trying to control infectious diseases. And also very recently, I've become interested with collaborators in combating the illegal trade of wildlife and in gene drive strategies for primarily for pest eradication. I guess the second characteristic is that the data is poor. And by this I mean that there's kind of a large amount of missing data in terms of the actual process that we're trying to study. So an example here, this picture here is trying to illustrate a possible chain of transmission of infection, say, within a household. So each of these lines here you can think of as an individual, where the yellow signifies the point at which they became infected, but there's a latent period, so they're not yet infectious, and at some point they become infectious, which we might assume kind of coincides fairly closely, say up to a day with the onset of symptoms, and at some point they recover, and you can see there's points here where individuals infect another individual within the household. So this is the kind of underlying process that's actually going on within this household, but what we're likely to actually observe in practice is just these days on which some of the individuals, and not all of the individuals in the household, show symptoms. And from this data, we need to extract information to try and understand and characterise the full process. This will be very relevant to the actual example that I come to in a little bit. I guess the third characteristic is that stochasticity and that we've got integer numbers of individuals, that interplay between that is very important. So the precise details of what's in this picture are not overly important, but it is, once again, modelling the spread of a disease within a population. And along the horizontal axis here, we have the number of susceptible individuals against here the log number of infectious individuals. The black dashed line there is if we used a deterministic model, so a model which didn't account for stochasticity, and you can see it passes through here and it converges in through damped oscillations towards a fixed point, which is endemic persistence of the disease in the population. Also plotted there is if we had an equivalent model that accounted for some stochasticity in the process, so in the timing of transmission events and whether transmission happens or not, the times at recoveries of individuals. And there was actually 5 realisations I did there, 5 different simulations. And we can see that because we're getting down to a handful of individuals here, it's possible that stochastic fluctuations means that you hit this absorbing boundary, no infectious individuals in the population, the disease has died out, and hence we'd require another introduction in the population to see disease dynamics. So it's important to account for this. You get vastly different outcomes and behaviours by accounting for this stochasticity. The final characteristic, I guess, of the research is that it is really a result of those various things I've just talked about. When looking at a problem of practical importance, needing to extract information from limited data and needing to account for stochasticity and integer dynamics, it becomes a computationally challenging problem to do this modelling. And so we look at developing methods which are computationally efficient to be able to achieve these. Okay, so on to now a particular example that I hope illustrates some of these characteristics that I've just talked about. And this is on characterising pandemic influenza. So if we look at the most recent pandemic of influenza in 2009, the swine flu pandemic, and we compare that to the 1918 pandemic, we can see that these are completely different outcomes, completely different beasts that we're dealing with. The 2009 pandemic, there was a large amount of asymptomatic infection, so individuals that were infected but didn't actually display symptoms, and there was moderate transmissibility. In 1918, there was very high severity, lots of mortality. And so in the early stages of the next pandemic, we need to know what we're dealing with in terms of its transmissibility and its severity. And so this is actually part of the Australian Health Management Plan for pandemic influenza. This is a figure taken from that plan, and along one axis we have looking at the transmissibility of the strain, broadly broken into low, moderate and high, and we've got the clinical severity, low, moderate, high, and here's some previous pandemics, strains that have been plotted there along with the ongoing influenza outbreak. And so in the very early stages we want to use the data we've got to try and characterise where we are in this space, and as part of the health management plan we also have a response that we might take based on where we are within this transmissibility severity space. The protocol that will be enacted to get data that we'll use to analyse this and try to do this characterisation is what's known as a first few hundred study. So following the first confirmed case of influenza in a household, we'll surveil that household and each day ask for symptoms onset within the individuals in those households. And so we'll get something like this, where each of these is a different household. We'll miss some of the cases, especially typically the first cases within a household, if we're going to miss some, because it requires them to show symptoms, to go to the doctor, etc. But once the surveillance is enacted, once we have a confirmed case within that household, we're more likely to pick up subsequent cases. And so we want to take this data and work out the transmissibility and the severity. And we've developed methods which do this. So here I've characterised transmissibility as a secondary attack rate. So this is the proportion of a household that gets infected in a typical, following a typical infection. And we've got severity here, which is a measure of how easily we can see and identify cases in the population. And this was done within it's a Bayesian method, so it's within a Bayesian framework. So we start with a prior distribution, which says we know little information about the transmissibility and the severity. And this is based on one realisation, so one synthetic data set. And we look at after 18 days of that outbreak, 25 days, 32 days, etc. And we look to see where we are in this space. In this particular synthetic example, the severity was 0.9, quite high, and you can see that we identify that very quickly, all the masses in this top row. And we also, the transmissibility was 0.55, which is in this middle region, but close to the next region. And we can quickly identify we're up in that region as well. So I can't go into the detail of the underlying methods that we've used, but they're at the kind of forefront of Bayesian computational statistics, and they involve consideration of the numerical and computational methods, and they also look at fusing a number of probabilistic techniques together to make them efficient and accurate. Just like to also mention some current related research that we're doing. So we're actually using this now to look at what resources the Department of Health will require in an actual pandemic outbreak, and how long we have to enact this to accurately or sufficient accuracy characterise the transmissibility and the severity. I'll also mention that we're working with the Australian Sentinel Practices Research Network, which is based at the University of Adelaide, which gets us very high resolution disease data from about 0.5% of GPs across Australia. And so this allows us not only to characterise seasonal influenza, which is of interest, but it gives us a baseline for comparison to pandemic influenza. So I'd just like to stop there, thanking everyone here, and I'd like to additionally thank my family, so my wife Ellie and my daughters Madeline, Phoebe and Harriet. Thanks.
Are there questions or discussion for Professor Ross? You gave an extraordinarily clear lecture. So please join me in thanking Professor Ross.
The 2017 Pawsey Medal recognises the contributions to science in Australia by Dr. J.L. Pawsey, a Foundation Fellow, Its purpose is to recognize outstanding research in physics by scientists up to 10 years post-PhD in the calendar year of nomination, and except in the case of significant interruptions to a research career. The recipient of the 2017 Pawsey Medal is Associate Professor Igor Aronovich. I invite Igor to come up to the stage now.
Associate Professor Aronovich is delivering breakthrough research that underpins next-generation light-based technologies spanning energy, communications and quantum information processing. His work is original, has motivated wider research and focuses on novel single photon sources, one fundamental building block in quantum information science. He has demonstrated new materials with record-setting properties which assist the further development of quantum communication systems and their deployment in real-world applications. His work contributes to one of the pressing issues in the modern era, ensuring that private information and sensitive data can be secured through unbreakable encryptions. Associate Professor Aronovich, congratulations, and I invite you to present your talk entitled Quantum Emitters in Flatland.
Thank you, Professor Riley. Thank you very much for the Academy for this great honor, and I would like to extend my congratulations to all the other awardees. So in the next few minutes or so, I would like to share with you the results of our group over the last few years and to talk to you about defects in solids and particularly in atomically thin materials. And I must admit, I stole this concept of flatland from the Nobel Prize winners Andrew Game. So when we talk about defects in solids, we of course most of us will think about two particular examples, and this is the blue hope diamond you can see here, and the blue here due to this boron atoms incorporated within the diamond, and of course the ruby. This is a sapphire or alumina, and the chrome inside will give you this beautiful red color. Now I should note that the value of this is of course by orders of magnitude more expensive. And what we're trying to do in our group is to try to isolate these defects on a single level. And what I mean by this is if we shine a green light or a green laser onto a material, it will only emit single photons, one at a time. And I will show you how we can measure this pretty easily these coming days. So to achieve this, we can actually explore quite a few different platforms. And this has been a significant research over the last almost 20 years. People were looking at diamond, at color centers in diamond, at silicon carbide, and more recently YAG and zinc oxide. And these materials have a very common characteristic. They have a very high band gap, which means that if you trap a particular impurity, let's say a chromium atom or a vacancy, within this high band gap material, you can create an artificial atom. So if you think about the physics that was done in the 60s and the 70s, where people were trying to trap atoms in this massive, big vacuum chambers, you don't need this anymore. You can just pick up a simple commercial diamond or a silicon carbide material and dope it with a particular impurity. And this will create this ground state and an excited state, and will create an artificial system that will emit red or other light sources. And there are several parameters that are important to consider when you want to design or engineer this type of system. And those include brightness, purity, repetition rate, efficiency, and stability. So really you want your source to have all this above techniques. All this above properties, apologies. So if you are successful in engineering those, you can actually think about technologies that are currently unavailable. And those include quantum communications, quantum computing, and probably some sort of sensing. And I would like, again, I borrowed these examples from the University of Chicago. This is an illustration of this atom, as you can see here, that emits single photons, some sort of a single chip futuristic computing. And this is an example of a sensing. It's some sort of a biomolecule that sits on top of a diamond surface, and underneath you have this source that emits single photons. And this is great because you can think that you can control and manipulate only one photon at a time, so you have access to polarization, to fluorescence lifetime, to intensity, and so on and so on and so on. But there is a major problem with these three-dimensional systems, the refractive index. And what it means is that the light that is generated within these materials likes to stay there. And so to eliminate this problem, we want to go to two-dimensional atomically thin materials. And in 2004, Andrew Game and Konstantin Novoselov received the Nobel Prize for graphene, in 2010, sorry. And you can think that graphene is a perfect example of this two-dimensional mesh of one atomic layer of carbon atoms, but there are more of those. And there is this nice review by Andrew Game that mention that you can actually stack them like a Lego. And there are different materials with different properties, and one example is this transition metal digital cogenite, and this material in particular is extremely bright, and you can even make a laser out of an atomically thin material. So the research that we are pursuing extends a bit beyond the single defects. But one particular atomically thin material that is very, very interesting is boron nitride. Now, boron nitride is typically a lubricant. It's a very cheap, dirty material. But if you can isolate them on a single monolayer, and I will show you how to do it fairly easily, you can actually generate this nice flake. So this is a schematic orientation, schematic illustration of these flakes. It is similar to graphene, but instead of carbon, it has boron and nitrogen. And you can generate single photon sources due to these defects. And that is what we did. So we started with this commercial boron nitride material, and you can see that you have a very strong luminescence somewhere in the red spectral range. And the defect that we think causes this luminescence is this anti-side nitrogen vacancy. So it is a nitrogen that sits on the boron side and nearby a missing atom. And this is an intrinsic defect that is very, very easy to make. And this correlation measurement indicates that indeed both the monolayers and the few layers are actually single photon sources. And I would like to steal another minute just to try to explain the physical concept, since the award is for the physical sciences. And to do this, it's actually very simple. You send your light through a beam splitter, and the only thing you measure is coincidences. You don't care about the intensity. You only measure coincidence. And if you do have only one photon arriving to your beam splitter here, then you cannot split a photon, and then only one detector clicks. And then if only one detector clicks at a particular time, you will have no coincidences. And that's what this graph shows you, that at zero delay time you have no coincidence, meaning that you only have one single photon source at a time. And so now when we have this source, the characteristics are quite fantastic. It's very photostable, as you can see here. It's very bright. It emits about 4 million counts per second that we can measure and harness these photons for practical applications. And it also has a very short fluorescence lifetime, which means that you can repeatedly generate these photons very, very fast. So it basically matches all the criteria that we want from a perfect single photon source. And we can measure some interesting properties. So for example, for the biological sensing applications, it's very important that the source will be excited with a longer wavelength to avoid any autofluorescence. And we can do it. We can shine, rather than a green laser, we can shine very strong pulses of infrared light and generate the same emitters at the visible. And you can see here, this is the infrared, and this is the visible excitation, and you can generate exactly the same single photon source. You can also put it on an optical fiber, the same fibers that connect our houses to the main computer network. And so here is, again, a schematic illustration. We have a HBN with a single photon source sitting in the middle of this fiber. And we can excite and collect the light with an objective, but we can also do a transmission measurement, excite from one end and collect from the other end. And you can see here, this is through the fiber, and this is through the objective. This quantum phenomena, this single photon generation, this magical dip, is maintained in both cases. So the system is very, very robust, and we can move it from one place to another. Now, I mentioned that it's very easy to make those in-house, and my team was very busy trying to do it, and we, of course, adopted, again, the same technique that Graham and Novoselov developed, using a simple Scotch tape. So we're doing the same thing. We're buying a boron nitride crystal and then just buying a lot of sellotape from different sources and exfoliating. And you can see that it works quite well. So in this particular example, you can see that this is a monolayer and a bilayer and a multilayer, and you can distinguish it just by using an optical microscope. And now, because we have large flakes that we can move from one place to another, we can think of even more advanced experiments. And in this case, we collaborated with Professor Terry Odom in Northwestern University to put these flakes on a metallic aperture that acts as a plasmonic hot source. So basically, you enhance your emission. And you can indeed see that we can get even brighter sources with even shorter lifetimes. So I would like just to summarize by showing you this cover that we just wrote a roadmap, that these 2D materials, that combination of 2D materials with the quantum properties and the optical properties are really the way to go over the next kind of 5 to 10 years to really achieve new systems for integrated computing, for new photonics effect, and for quantum cryptography. And none of this would have happened, of course, without the hard work of the team. And I would like to acknowledge particularly 2 students, Ton and Meran, who pursued most of the research that I showed you. Of course, I would like to acknowledge the Australian Academy of Science once again, and the Australian Research Council, and the list of collaborators from overseas. Thank you very much.
Are there questions or comments? That's another very clear presentation. Oh, there is one over there? Okay.
In Melbourne. Can you place these in series, sort of multiplex them and do equivalent of a Stern-Gerlach experiment with electrons?
Not yet, but we will try.
Thank you very much, Professor Haranovich.
The 2017 Ruth Stevens Garney Medal recognises research in human genetics and honours the contribution to science in human genetics by the late Ruth Stevens Garney. Its purpose is to recognise distinguished research in human genetics, including clinical, molecular, population, and epidemiological genetics and cytogenetics. The recipient of the Ruth Stevens Garney Medal in 2017 is Associate Professor Sarah Medland. I ask Sarah to come forward, and then Vice President Professor T.J. Higgins, Dr. T.J. Higgins will read the citation.
Sarah Medland is a statistical geneticist working on child and adolescent psychopathology and women's health. She was instrumental in the formation of the Enigma Brain Imaging Genetics Consortium, which is currently the largest brain imaging study in the world. Dr. Medland's work in this area has significantly advanced our understanding of the ways that genetics influences the structure and function of the human brain. Dr. Medland, congratulations. I call on you to present to us advancing our understanding of the genetic architecture of the human brain.
Thank you. And I'd like to start by, of course, thanking the Academy. It's a huge honour, and also in particular thanking someone who's been a great mentor to me, Professor Nick Martin. So in presenting this type of work, it is very collaborative. So the work that I'll be presenting today is from the Enigma Genetics Working Group, which I chair, and these are the authors from our most recent publication. Some of the many authors, I should say. So we founded the consortium in 2009, and it was really motivated by the fact that there was just insufficient power in the work that was being done within imaging genetics. So coming from a genetics background, we'd already seen this kind of thing happen in other traits, and we really wanted to try and avoid learning these lessons in public and to boot the field quite fast forward. So this is the email that went out, and I think it makes a really important point. Although our power calculations are often actually limited by budget, it is not actually meant to appear within the equation. So we're meant to think about sample size, effect size, and alpha, or our statistical level that we want to employ, but unfortunately it's often limited by cost. So we sat down and we made a list of everything that we thought had been problematic about previous consortiums we'd worked in, and then we tried to avoid that. It seems to have worked well so far. So at the moment, we're over 900 scientists from around the world. We have data and scientists from pretty much most places. And we work across many different areas with three main themes. So the work I'll be talking to you about today is about the genetic work that we've done. So these are meta-analyses, but they're meta-analyses of new analyses rather than meta-analyses of published findings. So as part of coming into the consortium, all groups agree to reprocess their imaging data, which is a substantial cost. And we do a lot of harmonisation via shared protocols and via QC. We make all of our protocols freely available online when we start a project. We know they are used by our competitors. We get a lot of emails from them. We try and help them out when they get stuck. Because we actually feel that making sure that everyone can do the best work they can is really important. We make our results freely available online as soon as they're published. From a genetics point of view, we're using genome-wide association. So we use these chips, which can very cost-effectively genotype a large number of probes that have been chosen to cover the genome really well. So under current budgets, you can get a very good genome-wide association for around $50 US. Obviously, when this technology started, it was a lot more expensive, but it's doing very well. We can then also use publicly available reference data and infer or impute in a lot of coverage because there is such a high autocorrelational structure within the genome. From the point of view of running these analyses, it's a fairly simple regression model. And we, of course, pay a lot of attention to known confounds. So how did it work? We put a call out to the literature. We had 22 groups, call out to the field. We had 22 groups respond. We ended up with over 10,000 individuals in our first discovery set. And in this work, we were looking at the total brain size and also the hippocampal volume. What we can say is that none of the groups individually would have had sufficient power to find anything, with the exception of the ADNI group, who rediscovered ApoE, but that's not very surprising because that's an Alzheimer's cohort. When we do this work, we use a high significance threshold to avoid false positives because the cost of having a false positive is very high. So taking forward a gene or a finding and then undertaking functional work when that location is not nailed down well enough or the effect size is not nailed down is a huge cost, so we employ a very stringent level. But bringing together all this data, we were able to actually discover the first robustly associated findings with hippocampal volume. These were very nicely replicated in a back-to-back publication that independently actually found the same hits. So we decided to have another go. We wanted to go bigger, we wanted to go better. We said we were going to look at the main subcortical structures. We put another call out to the group, and this time we had 50 takers. So, we established trust, we established methods, and then, we find that these things grow. So looking across the structures, I'm just going to highlight a number of results. Because of the way the imaging data is actually analysed, we usually correct for a total brain volume measure. This actually artificially creates a lot of orthogonality in our measures. So although when you think about the brain structure, you're expecting to see genetic effects going across structures that are right next door to each other, because we're actually making these global corrections, we end up removing a lot of that common variation. So when we look at structures, we're looking at hits that are really independent to that structure. What we were able to show is that the effect sizes we are seeing in the brain are no bigger than the effect sizes we're seeing anywhere else in the body. And this is incredibly important, as there's been a long history of hypothesizing that effect sizes in the brain will be much bigger. And unfortunately, most of the power calculations in grants and in sample recruitment are based on this assumption. So what we're seeing is within the brain, when you have a large enough robust sample, a large effect is around 0.5% to 1%. 1% is extremely large. If you contrast this with the previous hypotheses of sort of around 5 to 10% of the variation being due to a single genetic variant, you'll see just what a difference that is. Looking across the genome, we were able to demonstrate from the genetic data that all of these traits were significantly heritable. And just digging down into one of these, this is a putamen, which is a structure that's very important for motor and learning. This is a particularly nice example because we had a very strong effect over here. And looking at this data, we could see it was in a region with quite a few genes. So when we find this type of thing, the next thing we try and do is look at expression, and we try and see if the variants we're finding actually are associated with expression of any of the genes in the right tissues. So within Enigma, we're in a particularly good position that we can go ahead and look at this in replicates and actually get replication between different samples. What we're able to show is for that particular variant, it looks like there's a gene called connectin-1, which appears to be affected here. With shape analysis, we can also look at deformation of a structure compared to the usual atlas, and we can start mapping where on these gross anatomical structures this effect actually appears to be happening. This is important because different regions of these structures connect to different regions of the cortex, and so knowing where on the structure the effect is hitting enables us to sort of follow it downstream and have a look at what's going on. This work is not in the paper because we were actually asked by Nature to remove all our animal work from our human genetics paper. So we did that. But we had actually done quite a lot of animal work in model organisms and particularly in mice, and looking at the histology and what was going on. So what we could see from the work that we'd done was it actually looked to be influencing the shape of the cell. So variants in that we had found, they weren't really influencing the number of cells, but it was about the shape of the cell body as opposed to the other parts of the cell. And so this was having an overall effect on volume in these particular regions of the putamen. Okay, we have moved that forward, and what we're seeing is that the power is growing as sample size grows, as we would expect. Similar thing when we're looking at hippocampal volume. We're once again seeing increased power, increased signal, and we expect this to follow suit as we manage to get more samples for the rest of the brain. This is our current project. We're looking at the cortex. Here we are seeing, as you would expect, signals crossing these sort of artificial boundaries that have been placed on the cortex, and we are seeing a really nice sort of multivariate story coming together there with different genetic effects. That's unpublished, so I'm not going to show you the actual results. The other thing that we've been doing within Enigma is looking at case control meta-analyses, and we did this because when we went to the literature and tried to look to see whether these these regions were important for different diseases, we actually found a lot of heterogeneity, and so we needed to do phenotypic meta-analyses to try and sort these questions out. And same kind of thing looking across the lifespan. We actually found no population norms that would allow us to work out when we were QCing data whether the data we were getting was reasonable given someone's sex and age. So with that, I will end and thank everyone who's been involved in this work. I often get questions about what did everyone do. I can honestly say that everyone on this slide is absolutely essential to that paper. We lose one person, we lose a site, we lose a site, we lose power, we lose a hit. So it's collaborative work, but it's collaborative work in a really important way. So thank you very much.
Thank you, Dr. Medland. Any questions? Yes?
It's alleged that the corpus callosum in the average human female is 1.5 times as big in diameter as in the average male. And this enables the female to relate the two hemispheres much better than males can on average. Is the genetic source of that difference identified?
Interesting question. Corpus callosum is... I will start by saying corpus callosum is not an area that we have explicitly studied yet. I think there is quite a lot of variation in those findings. There is also different regions of the corpus callosum, and they connect different parts of the brain in different ways. So I think size is important, but size in certain parts of the corpus callosum is going to be more important. So I think I probably don't have an answer for you right this moment, but I think this is something that we could look at. Yeah.
Can we hear?
Any other questions or comments?
Up here.
Oh, there is one up there.
Yep.
Great.
Sorry, Joseph Getz, University of Adelaide. Congratulations, Sarah. I would like to ask you if you can comment why there was expectation that some of these traits, or there will be bigger effect size. Is that our simplification of how we evolved, or is it a reflection of that?
Great question. I think a lot of the misspecification, if I can call it that, of effect size has come from particularly candidate gene studies, where genes have been selected for study based on what's known about their role in a model organism or in response to drug treatment or pathology. A lot of those studies have had very small sample sizes, and unfortunately winner's curse has played a large role, and also the file drawer problem of non-significant findings not being published. So when you have a small sample size, for a study to get published, the effect size needs to be bigger. And I think if we could go back and actually gather all the data that was collected, we would actually find those effect sizes are coming down to, what we're actually seeing here. It's just we don't have access to that full data set to look at this.
Thank you, Sarah.
Thank you.
Thank you very much. I will now hand over to the President to present the Max Day Award.
Well, thank you, TJ. This is a really special occasion because we have a brand new award and we have the donor and family here to oversee it. This is the 2007 Inaugural Max Day Award, named in honor of Dr. Maxwell Frank Cooper Day, AO, FAA, who is the longest-serving Fellow of the Academy. He's present here in the lecture theatre, and he spent a lifetime championing entomology, conservation and forestry and helping other scientists. Elected to the Academy in 1956, just 2 years after the Academy was founded, Dr. Day still clearly remembers the 1958 ceremony at which the then Prime Minister, Mr. later Sir Robert Menzies, officially laid the foundation stone of what is now the Shine Dome. And the quiz question is, because I couldn't answer it when I was asked, where did that stone come from? Dr. Day will know. Dr. Day said recently that the most satisfying achievement of his long career was the work he did with the late Professor Frank Fenner, AC, FAA, FRS, on developing myxomatosis virus, which played a major part in controlling Australia's rabbit problem. Dr. Day is also proud of his work establishing the Division of Forest Research within CSIRO. And just a personal note there, Dr. Day, I think you're an exact contemporary of my father who worked in the Division of Building Research. So it's very nice to have that overlap. And that led to Australia's international recognition for its contribution to forest research. Dr. Day, his family, Mr. John Day, and Mr. Doug Hooley have generously funded this fellowship to support scientists early in their career, acknowledging the support that Dr. Day himself received as a young researcher for travel overseas to gain his PhD at Harvard. The award for the Environmental Science Fellowship is a certificate, a very fine certificate, which you can see if you like. Dominic here has a copy there, which is the certificate in the scroll, and then a fund up to $20,000 to assist PhD students or early career researchers to meet costs associated with their research or to supplement PhD scholarships, as well as an acknowledgement of exceptional research proposals through the presentation of the certificate. The highly competitive award received 110 applications, and I'm pleased to announce that the recipients of the 2017 Inaugural Max Day Environmental Science Fellowship Award are Mr. Nicholas Laserberg and Dr. Marta Yebra. Now, we're going to do it this way, just so everybody knows. There are going to be 5 recipients. There are 2 for the Environmental Fellowship and 5 highly commended recipients. And I'm going to stand over there to take a photograph and shake their hand, and then they are going to shake Dr. Day's hand as they return to their seats. Now, I have the first certificate with me, but Dominic's going to bring the others over to me. So I'm going to move over now. I first announce it, and then I'll move over. The first recipient, we're using alphabetical order to name the recipients. The first recipient is Mr. Nicholas Laserberg, and he's receiving his award for his project entitled Ecology and Conservation Biology of the Night Parrot. So, Nicholas, I'm going to go over here so we can take. I need to press it yet. I'll stay here now, and I'll now ask Dr. Marta Yebra to receive her award for her project entitled Flammability Warning from Space. Now we have these three highly inaugural Max Day Environmental Science Fellowship highly commended awards. And they're going to be, and I'll call them out in order, Dr. Hugo Harrison, Dr. Carenza McElroy, and Mr. Max Worthington. So in alphabetical order, I will now invite Dr. Hugo Harrison to receive his award for his application entitled Managing Australia's Coral Reefs for Recovery and Persistence. Hugo. And the next recipient is now, I invite Dr. Carenza McElroy to receive her award for her application entitled The DNA Footprint of Near Extinction, Interrogating 100 Years of Black-Throated Finch Decline by Sequencing Contemporary and Historical Specimens. And now I invite Mr. Max Worthington to receive his award for his application entitled Renewable Polymers for Agriculture and the Environment. Hand over to Anna Maria Arabia, our Chief Executive, to tell you about what's next.
Thank you, Andrew. I'll take the occasion to thank Dominic Burton for all of his work over the course of the year in managing all of the awards. Thank you very much, Dominic. You do a superb job. Before we break, could I ask all of the awardees to please make their way to the front entrance of the Shine Dome for a group photo. We'll also be taking individual photos of early career awardees and a group shot of all awardees, followed by the benefactor awardee lunch, which will be held in the marquee just outside of the building. Lunch will be served in the Jaeger room. The closed session of the AGM for fellows will commence here in the theatre at 2:30 p.m. Before we go, photos of fellows of the Academy of Science. If you would like your photo updated for our website, please make your way to the foyer, and Jen Van Dyke will be there to take a current photo of you. We're very happy to do that. The Early Career Researchers workshops will also commence at 2:30 p.m. There are 4 of them. Storytelling for Researchers will be held at University House, just down the road. How to pitch your research to the media will also be held at University House. Those of you who are registered for these workshops, please meet no later than 1:50 p.m. in front of the Shine Dome, and a member of staff will take you to the venue. The third is Fostering Partnerships with Industry, and that will be held in the Becker Room on the ground floor of the Shine Dome, so just turning left if you exit from that door over there. And the fourth, Perfecting Grant Writing, will be held in the ground floor boardroom of Ian Potter House, just on the other side of the car park. I hope you've all had an enjoyable morning and afternoon, and I look forward to seeing you all at dinner tonight at the Arboretum, which begins at 6:45 p.m. with a pre-dinner cocktail reception. Buses will leave from the QT Hotel and University House at 6:30 p.m. sharp. Just before you leave, I'd like to give you just a short snapshot of the symposium that will be on tomorrow, and I encourage as many of you to attend. The symposium is entitled Life on the Loose, Species Invasion and Control, and here is a short peek of what's to come.
We really didn't understand just how rapidly it was going to expand. And that's the food that many species depend on.
Thank you. We look forward to seeing you tomorrow, and for fellows this afternoon, please enjoy your lunch.